Welcome to today's live webinar supported by AstraZeneca and MSD. Today we're going to be talking about variant classification and interpretation and use of guidelines and databases. Next slide, please. So this is one of a number of masterclasses that we're running across the course of the year, and they're designed to support the EQA schemes that we're running around variant classification. I'm Simon Patton. I'm the Chief Executive of EMQN CIC based in Manchester, and with me is my co-chair, Professor Sandi, the Director of GenQA in Edinburgh in the UK. Next slide, please. So a quick overview of our agenda today, just to give you a sort of a flavour of what we're going to be covering. After our introduction from myself and Sandi, we're going to be covering the overview and application of ACGS guidelines for classifying ontogenicity of somatic cancer variants and recommendations from the UK Somatic Variant Interpretation Group. And then we're going to move across to multifactorial genetic epidemiological studies and the use of likelihood ratios for variant interpretation. Finally, and most importantly, these webinars are designed to be interactive. The educational component is a key part of what we're trying to do. So please do interact with us in the live question and answer session at the end. We've got about 25 minutes allocated for it. You will see at the bottom of your browser bar, your Zoom bar, that there is a Q&A option. Please submit your questions through the Q&A. I'm sure Sandi will cover that very shortly in the housekeeping points. And without further ado, I'm going to hand over to my co-chair, Professor Sandi Deans. Sandi, over to you.
Thank you, Simon, very much. So if we move on to the next slide, please. So Simon and I today are joined by an excellent faculty. We are very delighted to introduce you to Dr. George Burgall, who is a consultant clinical scientist and the deputy leader of cancer genomics in Manchester as part of the Northwest Genomic Laboratory Hub. And we also have great pleasure in introducing Dr. Charlie Rowlands, who's a bioinformatician based at the Institute of Cancer Research in London. Next slide, please. So just some housekeeping before we start with our presentation. So we've muted your microphone to avoid any background noise. The webinar is being recorded, and the reason for this is so that we can then, once it's gone through compliance, we can make this webinar available to you on both the GenQA YouTube channel and the EMQN YouTube channel. So it's a great resource for learning for yourself and other colleagues. And as Simon says, that we do want to hear from you and have some interaction. So you can use the Q&A function that you should see on the bottom of your screen to submit questions at any time during the webinar. And then we hope to cover as many as possible as we can at the end of the session. Next slide, please. So we do like to deliver these webinars to meet the needs of all of our participants. So we do listen to your feedback, and it's incredibly important to us. So we really would like you to complete the a short feedback survey. It shouldn't take you very long. After the webinar, you'll receive an email, and just click on the link and you'll be able to give us your feedback there. As I said, couple of minutes and details will be shared at the end of the presentation, but we really do appreciate you taking the time to do this so we can actually make sure these webinars are focused to the areas that you would like us to discuss. Next slide, please. This is a non-promotional webinar organised and funded by AstraZeneca and MSD, and these are the disclaimers here. Next slide, please. So, as Simon mentioned, there's two areas that we're going to focus on today. One is about the overview and application of the ACGS, so that's Association of Clinical Genomics Science in the UK, for classifying both the oncogenicity of somatic cancer variants and really bringing in the recommendations of the published UK Somatic Variant Interpretation Group, what's also known as SVIG. We also are then going to move on to more of a bioinformatician's point of view about multifactorial genetic epidemiology studies and the use of likelihood ratios for variant interpretation. And this is about covering integrating the ACMG and AMP guidelines with curated databases to assess variant evidence and refine that classification. And also, how do you communicate the clinical significance in the context of multifactorial genetic epidemiology studies for variant interpretation? So a lot of really incredibly useful information is going to be shared today. So next slide, please. So it gives me great pleasure now to hand over to George, who's going to kick us off and tell us around how we can apply the guidelines when it comes to somatic variant interpretation. George, over to you, please. Thank you.
Thank you very much, Sandy, and thank you, Simon, for the introductions. Hi, everyone. I'm going to focus to start with on somatic variant interpretation guidance. Now, I'm very aware that a lot of the people who join this webinar work more on the germline inherited side. So the plan is to just give you an overview of the somatic guidelines, go briefly through the different criteria that are available, and highlight where things align and match with germline setting and where things can differ. So as you all know very well, cancer testing is expanding very quickly, and we're using larger and larger panel, including whole genome sequencing in cancer diagnostics, both for solid tumors and hemato-oncology. And when we started this work back in 2018, we realized that there was a variation in the way at least UK labs addressed somatic variants in the way we classify them and in the way we report them. And we know that was an issue across different places around the world. And one of the main reasons is when variant interpretation guidelines started to be published in a very structured and standardized manner, like the ACMG, ACGS, and CAMVIG, they were focused on germline side. They were not designed for somatic variants. So when we started, we wanted to establish a consensus approach to somatic variant interpretation that is tumor agnostic. Now, while we were working our way through this process, more than one framework or guidelines have been published. There was the Freyen et al., the Belgian guidance in 2019, and there was also the Horak et al. In 2022. So we expanded our work in order to review these different guidance and frameworks and try to adapt them to create an inclusive and standardized interpretation approach. So this guidance that I'm going to present was published last year in the Journal of Medical Genetics. It's open source, so you can all download it, but it's also available on the ACGS website under the best practice guidelines, the variant guidelines section. You can download the document and the supplementary material in there, and the links are available. And what I'm going to do is I'm going to give an overview of these guidance and how we structured them and reflect, like I said, on any similarities and differences with germline setting. So one of the guidance that many of you may be familiar with is the Horak et al. 2022 guidance, which basically published a somatic guidance that really aligns with the ACMG germline criteria, presenting a points-based system for oncogenicity assessment. And while we were reviewing the available guidance, we decided to adapt the same oncogenicity pathway. So we took the ACGS framework. I know you can't see my mouse, but it's the right-hand side of the screen, and adapt that with a slightly different terminology to clearly show the referring clinician that this is a somatic setting. And so we moved over from pathogenic and likely pathogenic to oncogenic and likely oncogenic. Benign and likely benign remain the same. We don't report them anyway. And so we reviewed Horak, we adapted that, and we aim to bridge some gaps with other guidance. We expanded the strength flexibility in which you can apply criteria, and we also introduced novel aspects that we will have a look at. And all of this work is focused mainly on oncogenicity. So that's the biological impact of the variant in the tumor, and it is designed to be used alongside the AMP criteria on actionability. So that's Lee et al. 2017. So this is an overview of all the guidance. You can see we have 7 benign criteria and 11 oncogenic criteria. Any benign criteria is starting with a B and just numbered in order of how we presented them in the guidance, and same for oncogenic criteria, they're the letter O and numbered. And you can see an important difference from the germline criteria labeling, the current germline criteria labeling, which we expect to change by the end of this year or early next year. We haven't added a second letter to cover the strength of evidence because, as was evident after the publication of ACMG, all criteria started to expand to cover the strength of evidence. So you have your criteria and you have your strength of evidence depending on how you can apply it. And another important thing to highlight when you're dealing with a somatic setting compared to a germline setting, when we're reporting germline cancer genes and reviewing, classifying these variants, we're mainly dealing with tumor suppressor genes, BRCA1, BRCA2, MMR, APC, and so on. While in tissue testing, in somatic testing, we're testing variants or reviewing variants in genes that can be oncogenes, where they function through, where they are gain of function impact, or tumor suppressor gene where is a loss of function impact. And that does change the application of different criteria, as we will see. So one of the important things to get familiar with, which I'm sure a lot of you here will know about, is the cancer census list. That was the original publication in Nature Reviews, the census of the human cancer gene, and there was then a follow-up Cosmic Cancer Gene Census publication 2018. The lists are available from OncoKB or from Cosmic if you have a subscription with Cosmic, but basically it lists the gene and the known mode of action, where it is oncogene or tumor suppressor genes. Of course, that's not always straightforward. There are there are genes where they can play different modes of actions in different tumor types, and so it can be complex in certain instances, but for many of them you can see clearly whether it's an oncogene or a tumor suppressor gene. So whether you're looking at a gain of function variant impact or loss of function variant impact. Now we'll start with the criteria, and like I said, I'm not going to cover them in details. I'm going to give an overview and highlight some key differences, which will hopefully give us time for questions at the end and give us time to listen to Charlie's talk, which I'm also very excited about. So the first criteria that I wanted to present is O1. That's a criteria that does not have a germline equivalent. It is basically what we call canonical variant. If you see a variant that is on a specific list that I'll present now, then we are recommending that you take this as a standalone oncogenic or likely oncogenic. Now, an important thing to remember in the somatic setting, scientists are dealing with a much higher number of variants per patient or per case, and also under a much shorter turnaround time. So one thing that's important to try and address when you're trying to ask people to follow a very structured variant classification pathway is to help them to try and provide means that will help in speeding up the process. This idea was first presented by the Fryan et al. It's not present in HORAC, so we were inspired by the Belgian criteria, and we presented a set of variants that we called canonical variants that meet the criteria for O1, and we set certain criteria for it. So they have to achieve oncogenic classification. Most of them, there are a few exceptions that only achieved likely oncogenic. They should not have any contradictory piece of evidence, and they need to be highly recurrent and enriched in tumor databases. So we collected many variants from the SVIG group and multiple other labs, and then all these variants were reviewed and agreed by at least 2 SVIG UK scientists, and we published them in the guidelines as 2 lists for solid tumors and hemato-oncology, and we're recommending if you see it, you can use the classification and you don't need to worry about it. The next criteria is O2/O2 RNA. This is an equivalent of the very familiar, hopefully, PBS1 RNA based on Patton et al. And also importantly later on Walker et al. This is a null variant in a tumor suppressor gene. So, and this is key here. This is one of the first areas where you want to know that the gene you're looking at is a tumor suppressor gene. So if you're not familiar with it, go look at the gene in the cancer census list. And if so, and you're identified a null variant, a nonsense, frame shift, canonical splice site, etc., you can apply following the detailed guidance ranging from supporting to very strong as per the principles of Patton et al. And Walker et al. The next criteria is O3 and B1. So these are the germline equivalents of PM2 and BA1 and BS1. So this is talking about the presence or the absence of a variant in population database. O3 is equivalent of PM2. We're still allowing the use of both supporting and moderate. We wanted to remain in line with CanVig, but within the strict application that it has to be 0 observations across all populations in NOMAD version 4.1, including the UK Biobank. Hopefully reducing the times it will be used, and we will be reviewing the application of this criteria in the future. B1 can be a standalone benign if the variant is present in greater than 1% in the population, or if there is an expert panel group recommendations for any of the genes and greater than 0.1%, you can apply it as a strong. B2 is a criteria that doesn't have a germline equivalent, and this is a variant that does not fit the mode of action. There are some parts of germline guidance that are equivalent to this, but basically, if you identify a variant that does not fit the mode of action of the gene that you're investigating, you can go straight away to standalone VUS. And what we're recommending, you don't have to worry about doing more criteria. Again, this is with the aim of speeding up variant interpretation in a somatic setting. And one of the easiest, most common applications of this is if you see a null variant, a truncating variant like we looked at earlier, but in an oncogene. So you identified a nonsense, a canonical splice site that's resulting in a frame shift and termination. It is an oncogene. You wouldn't report that straight away VUS. You don't have to worry about doing a detailed classification, and you can move on. Next criteria is O4. This could be considered similar to PS4 case control analysis in germline setting. However, it's not a case control analysis because of the difficulties and the complications of having a denominator from these tumor databases at the moment. But essentially, is this variant enriched in somatic variant data basis? And if it is enriched, depending on the variant type and the tumor setting that you're looking at and the database that you're using, you are able to apply different levels of strength. And there are multiple resources that you can use to look for enrichment in databases. One of the most commonly used and one of the largest resources is CBio portal, where you can have a wealth of information on the enrichment of a variant in different tumor types. Of course, Cosmic, if you again have a subscription with Cosmic, that's another resource which can provide information on tumor enrichment for many tumor types and many genes. The next one is O5. This could be the equivalent of PM5, again, if you're familiar with those. And that's a variant that affects the same location and or result in a similar impact. So this covers splicing variants. This can cover missense variants we take into consideration. The other variants, what they're classified as, how do they compare in terms of in silico predictions, for example, Revel, if you're looking at a missense, and so on. We also reviewed the Walker et al. In this sense, and applied very similar principles to our somatic codes. So O6, O2, and O5. So we created, based on Walker et al. For the splice site variant, a very similar table that covers the different combinations to be able to apply this code for variants that you're investigating against reference variants that affect the same region, the same splice regions, and be able to use the combinations as was recommended by the Klingen SVI splicing group. Then O6 and B3, these are your in silico predictions tool. O6 is for oncogenicity, and BP4 for B3 for benignity. So you're again using the standard computational evidence, Revel, Splice AI, and so on. Now, important to realize here that you need to be a little bit more careful when applying Revel scores for gain of function. So for loss of function tumor suppressor gene, you can apply it as per the cutoffs that are listed. But for gain of function, what our review of the literature showed that they're not as well established using these in silico scores. So if you're looking at an evidence towards oncogenicity, you can apply the Revel score if you're looking at a variant within a known functional region where the missense variants are known to result in gain of function. However, on the benign side, we're recommending not to use Revel scores as an evidence against pathogenicity or oncogenicity as evidence towards benignity, because it is not as well validated to predict that. So another important area where you need your distinction of tumor suppressor gene, oncogene, and the mode of action in the tumor type that you are reviewing. O7, this could be equivalent to PM1 and PP2. We're talking here about if the variant is located in a mutational hotspot or a well-established functional domain. And for those working in a somatic setting, you'll be very familiar with the Memorial Sloan resource of the cancer hotspots. So we recommend different cutoffs for your variants. If you go on the cancer hotspots, you look for your variant and the gene, and you're able to see how enriched this as a mutational hotspot, and then you can apply the evidence at these different levels. Then O8 is the overall constraint for the missense variation at the level of the gene or a domain or a region that could be used where missense variants are known as a common mechanism of the disease. In this case, it's equivalent to PP2, and you can find a lot of these constraints, for example, on NOMAD, where you have the Z score, and you can apply it that way. Then you have B4 and B4 RNA. These are the synonymous or intronic variants where there are no predictions, or the predictions give no impact on splicing. You're able to apply that at different levels, or where you have the RNA evidence supporting no impact. So this is again for variants that could result in an impact on splicing, whether they're synonymous or intronic variants as well. O9 or B5, these are variants that could affect the protein length through in-frame deletions or insertions. If you have a variant, an in-frame deletion or insertion affecting a non-repeat region and resulting in a single amino acid change, that could be applied at a supporting level, and if it's impacting and resulting in a multiple amino acid length change, that could be used at a slightly higher level of moderate. And of course, if it is appearing in a repetitive regions with unknown function, then that could be used as evidence towards benignity rather than oncogenicity. Then O10 and B6, this is the criteria where you can apply your functional evidence. So if you have a well-established functional studies demonstrating a functional impact on your of the variant, you can use them at a very expanded range, from supporting to very strong in an oncogenic setting and supporting to strong in a benign matter. Now we know in a somatic setting, this is one of the criteria that we get a lot of feedback and questions on. What we recommended is the application of the Bernick et al. Criteria that was published a few years ago in the Genome Medicine journal for how to structure PS3 and BS3. This is the equivalent germline code, because one of the reviews that ClinGen did identified that application of functional study could be very subjective, and so it was resulting in a huge variation in classifications because different people applying the functional studies at very different strength. So what they set in this framework is a very specific criteria to assess your functional studies in a way to allow the use of a tiered strength evidence that is supported by a statistical analysis, giving you a confidence odds of pathogenicity in the strength of your study. Now, a lot of the studies that are published are on the well-established genes like BRCA1, BRCA2, P53, and so on. I've presented last year on the use of CANVAR, and there's a brilliant resource there on accessing a lot of the functional studies. But people struggle with it because a lot of other cancer genes, and especially sometimes the oncogenes, which again haven't been assessed in a germline setting, don't have as big functional studies. So people have been feeling that they're missing out on a pieces of evidence because of this criteria. But importantly, with the advent of MAVE studies, the large multiplex variant effect studies, there will be more and more studies published covering also oncogenes that will hopefully make the use of this criteria more efficient. But we did feel that it was necessary to follow the Bernick framework and recommendations, which hopefully will standardize the approach and make sure that the functional evidence is only used when it's worthy to be applied at these strength levels. The final main criteria of oncogenicity and benignity is O11 and B7. There's no such germline equivalent for this, and this is one of the criteria that's not been present in any of the other guidelines, is the tumor phenotype. So this is where you're able to apply evidence that is, for example, from other studies. When a tumor is reported, or they can go a lot of non-genetic analysis. For example, other than functional studies, they may have had immunohistochemistry analysis, for example, FISH, or other studies that can be gathered together to create what we call a tumor phenotype. Of course, that also could contain genomic signatures like tumor mutation burden, or CMV analysis, and also a patient phenotype. These together, we try to set different criteria where you can combine them with or without the germline knowledge. A lot of the, for example, the MMR analysis we do in a tumor setting is done on patients where they've excluded germline reasons for it, which may be a little bit different from when you do the analysis on a sample where germline cause hasn't been excluded, and so on. So we present different criteria. Some of it is detailed, like you see in this table, and some of it sets out the principles of where we are advising the use of what we're calling a tumor phenotype in this sense. And similar to the CANVIG guidelines, to avoid double counting or over using certain evidence, we created somatic specific tables for combining evidence covering both oncogenic criteria and benign criteria to reduce evidence double counting and make sure that we are not over interpreting our variants. And importantly as well, we understand that a lot of, as the genes that we are testing for in a somatic setting are expanding very rapidly, we are coming across a lot of genes where there are established germline guidelines, such as BRCA1, BRCA2, and the mismatch repair genes. And what we recommended here, because some of these genes have a very high likelihood of being germline in origin, even if you identify them in a somatic setting, and there's a lot of established and published evidence, including evidence that can't be easily applied within somatic setting, for example, segregation analysis and family studies. We allowed, or even in certain circumstances would actually advise on the use of constitutional guidance for some of these genes, such as BRCA1, BRCA2, and the MMR genes. Although, on the other hand to that, there are genes where the germline criteria can be too strict to be adopted into somatic setting. For example, P53 published clinician criteria will require a lot of phenotypic evidence in order to reach to a classification of pathogenic in a germline setting. But in a somatic setting, the likelihood of a P53 variant of being oncogenic does not need to meet as many criteria. So there are genes where we recommended the use of the criteria from a germline setting, but there are genes that is left for the local policy or local lab to assess whether they want to apply the constitutional guidelines or they want to use specific lines of evidence from the constitutional guidelines. Of course, that's different when you have a variant that you are concerned about it possibly being a germline in origin, then that comes under a different set of criteria where we would recommend that you classify the variant using germline guidance before you report it being at risk of being germline in origin. And finally, to support the release of these guidelines, working with Chris Watt, one of my colleagues in Northwest GLH, an excellent GTAC practice educator, we devised a set of educational materials. Chris worked on producing a lovely set of educational material that was reviewed and approved by the ESPIC scientific group, that contains worked examples of variants, tutorials, video, and written material, and various advice and tips, including practice variants and answers to them. They're currently available also on the ACGS website, same link for the guidance just underneath that. I'd highly recommend using them. And for anyone who's doing training in their lab, if you need the results criteria, you can email me or Chris to ask for the password for this material, because we're trying to protect it to make sure that people are assessing themselves objectively. This is just a presentation part of that educational material, which goes very nicely through the different criteria and presents them in a very efficient and easy to understand manner. And finally, I would like to thank many people who have worked so hard on producing the ESVIG criteria, and also again Chris for his contribution to the educational material, and we were supported by many organizations along the way. So thank you very much. That's the end of my slides.
Thank you, George, very much indeed. And now I'll pass over to Charlie, who's going to talk about our multifactorial genetic epidemiology studies. Charlie, over to you, please. Thank you.
Brilliant. Thank you, Sandi. So hello, everyone. I'm Charlie. I'm a bioinformatician at Claire Turnbull's team at the Institute of Cancer Research down in London. I'm very excited to speak today about multifactorial genetic epidemiology studies and how we use these in variant interpretation. So this is very much a shift into the purely germline sphere compared to George's talk. Let me just check I've got control of the slides. There we go. So I'm going to just go back a little bit to basics here, because I'm sure lots of you have used genetic epidemiology studies in your day-to-day work, especially in the context of BRCA variant interpretation. But I think we take for granted the amazing power of these studies and the maths that underpins them. So just have a quick introduction to what a likelihood ratio is and how they apply to variant classification under the ACMG guidelines. Then go into some of the basics of what these GenEpi studies are actually doing, talk briefly about how we implement them clinically in terms of guidelines, and then talk a little bit about how we can use databases such as Canva UK, which is a database administered by our team, to access these data in an efficient manner. I don't need to preach to this audience, I'm sure, about the ACMG version 3 guidelines, but these were released in 2015, and they were essentially the first big effort to really systematize how we conduct variant classification. So in the original formulation of the framework, we had these discrete categories of evidence that were assigned a relatively arbitrary level of evidence, strong evidence towards benignity all the way up to very strong evidence towards pathogenicity, as we've seen with George's SFIG framework. That sort of trend is still how we operate nowadays. And so this was designed very much by expert groups, but a lot of these placement of evidence items was not necessarily based on evidence itself. It was expert consensus as to how strong a particular evidence item should be applied. And critically, at least in the original formulation, we were only really able for most evidence items to apply the evidence at one fixed strength. So you can see there's exceptions for segregation data and for population frequency data. But by and large, there was a static level of evidence applicable for a given evidence item. And similarly, the sort of combining criteria, again, were logically informed, but relatively arbitrary as to how you decide what the final classification for a variant is based on the constituent items. So, we decided collectively that one very strong and one strong item was enough for a classification of pathogenic, but that one very strong and one moderate was only likely pathogenic, and so on. So there was a degree of arbitrary decision making involved in the generation of these guidelines. Then in 2018, Sean Taftigian, as part of Les Bieseker's group, in collaboration with Les Bieseker, released this Bayesian reformulation of the ACMG framework. And the key thing here is that we moved from a categorical approach to actually an empirically quantified approach, whereby we use this value, the likelihood ratio. And if you can determine a likelihood ratio for a piece of evidence, that can mathematically dictate what level of evidence is appropriate for that evidence item in that scenario. And so it was discovered through lots of maths that you can look at in the original publication that 2.08 is the magic number for these likelihood ratios. So 2.08 is a sufficient likelihood ratio to get you to supporting evidence. If you square that number, you get sort of 4.2-ish, I think. That's sufficient for moderate evidence. If you square that again, so 2.08 to the power of 4 is equivalent to strong. And if you square it one more time, 2.08 to the power of 8 is very strong. So critically, we now have a sort of mathematical yardstick by which we can determine the applicable evidence for a particular evidence strength for a particular evidence item. And then all you have to do at the end is you multiply your constituent likelihood ratios for each of your pieces of evidence, and that gives you a final classification strength. And so critically, if you can calculate a likelihood ratio for any given evidence item, you can then map it back to an evidence strength. And what this means is you can potentially have multiple evidence strengths for a single evidence item. So if we think in the context of in silico tools, if people are familiar with the Pajava et al. Paper from a few years ago, we went from being able to apply in silico data at one strength to expanding the number of different evidence strengths that a single in silico tool can provide. And we can also do gene-specific adjustment of evidence strengths. So certain evidence items are much more pertinent for some genes than others, and we can actually quantify that in a gene-specific manner. So it really allows much greater diversification and expansion of the original version 3 guidelines. This is just a very quick table to show how the different likelihood ratio values map onto the equivalent evidence strength. you'll see that these have a number of names. So they're either likelihood ratios, likelihood odds, odds path, Bayes factors, as we'll see in a few slides' time. So there's lots and lots of different names for precisely the same thing, which is this quantitative measure of applicable evidence strength. And then in more recent years, we've moved towards the idea of evidence points, which are actually simply the log base 2.08 of your likelihood ratio. And these are additive rather than multiplicative. So you can take all of your constituent evidence items, convert them to evidence points, and then add them together to get a final classification. And you can see that these map nicely back to your evidence strength. So +1 is supporting, +2 is moderate, and so on. And working in this exponent point framework allows us to combine, sort of determine all the combining rules that were previously sort of arbitrarily decided during the drawing up of version 3. So we can see that a very strong evidence item which gives you 8 points and a strong evidence item which gives you 4 points gives you a particular exponent sum. And that corresponds to a classification of pathogenic. And we can sort of start to fill out more systematically the different combinations of evidence items and the evidence strength that they should correspond to. And so this likelihood ratio based approach by which we can generate sort of bespoke evidence strengths for particular evidence items has been very widely adopted. So this is just a selection of different publications looking at different evidence item codes. So you can see within codon concordance, people have quantified how much evidence towards pathogenicity or benignity PM5 gives, specifically in BRCA1 and MSH2. We've got the Pajava paper in panel 3, that in silico one. George has already alluded to the Brinich framework, which is in panel 2 there. So this is now being widely adopted as the sort of way to dynamically interrogate your evidence items and your evidence strengths. And this is going to be the currency really of ACMG version 4 as well. We're moving away from the categorical approach where everything has to be either, you know, supporting, moderate, strong, very strong, to a numerical system that's based on these exponent points. And so what are these likelihood ratios? So these all operate in the field of Bayesian statistics. And in Bayesian stats, what you start with is a prior expectation that your variant is pathogenic. So if there is absolutely no evidence provided and a variant comes to you through diagnostic testing, what is the background likelihood that it's pathogenic before you've even looked at all the evidence? And then at the end of your calculation, you get a posterior probability of pathogenicity, which is essentially your final answer, your estimate to how confident you are that it is pathogenic. And what connects them is this value, the likelihood ratio. Like I say, sometimes these are called Bayes factors. If you read anything that's got a sort of mathematical leaning to it, any kind of statistical publication, you'll often hear them called Bayes factors. And all it represents is the difference in likelihood based on your new evidence that a variant is pathogenic rather than benign. And so as a formula, it looks like this, and it's translated in slightly more friendly language to the likelihood of observing your evidence under a model of pathogenicity divided by the likelihood of observing your evidence under a model of benignity. And to quantify that, naturally, what you need to do is generate two models. You need to, for a given evidence item, generate a model of how you expect that evidence item to behave under a pathogenic assumption or the assumption of pathogenicity, and how you expect that evidence item to behave under the assumption of benignity. So this is a really oversimplified example of how one might do this. So this is using the example of an in silico tool that's been released, and how you might determine the likelihood ratio for a particular score in this newly released tool. So what you could do, for example, is map out all of the in silico scores for this new tool for pathogenic variants and say, okay, if I have a newly identified variant that has a score of 0.8 on this assay, what is the likelihood that it is pathogenic? And if you follow that purple line of x equals 0.8 up to your curve, you can see that the likelihood, which is equivalent to the density on this plot, the likelihood is 0.302. And then we also need a model of benignity. So you do the same again, but you map out all of the scores for a set of known benign variants and see what likelihood your 0.8 score is of fitting the benign distribution. And so we have these two competing models, one of pathogenicity, one of benignity, and we look at the relative likelihood that a particular item of evidence, in this case a score of 0.8, is indicative of each. And in this case, and like I said, this is a gross oversimplification, and you actually would never do this if you go and look at the Pajava paper. It's far more complex. But a sort of back of the envelope calculation might suggest that we would get a likelihood ratio of 2.27. So the likelihood that the pathogenic model fits the evidence is about 2.2 times higher than the likelihood that the benign model fits the evidence. And so that could then be converted to exponent points by taking log to the base 2.08, and you would get a exponent point just over 1, so just over supporting. And so, as I've alluded to, if you have independent evidence items, you can combine their respective likelihood ratios by multiplication, and this would give you something equivalent to the odds path, the total odds path for that variant. More commonly now, we have moved into this log likelihood ratio space, which is the same as the exponent point sort of space, where you take the log, and then once you've taken the log of your constituent likelihood ratios, you just add them together to get your final total evidence points or total log likelihood ratio. And so how does all this relate to multi-fac studies? Well, what we can leverage is the fact that, and we're now talking in the context of BRCA for the rest of this webinar, but we can leverage the fact that a lot of characteristics of the clinical features of a proband or the sort of pedigree, family history factors of a proband are very distinct between individuals who have a pathogenic BRCA variant and those who don't have a pathogenic BRCA variant. And so we expect individuals with pathogenic variants to have far stronger family history of relevant cancers than individuals without BRCA variants. There's various pathological and histopathological features that are very distinct in BRCA, or certainly far more prevalent in BRCA carriers than non-BRCA carriers. And then there are things related to the variant, the sort of behavior of the variant itself. So we expect the variant to co-segregate with disease in families where the variant is causative of disease. We would expect in the context of BRCA1 and 2 specifically that should be minimal co-occurrence of pathogenic variants with a second pathogenic variant. So you might have a benign variant that occurs in trans with a pathogenic variant, but you shouldn't have a pathogenic variant occurring in trans with a pathogenic variant. We can also leverage case control signals. So that's a much more sort of only 1 or 2 studies have tried to evaluate the likelihood ratio for case control data, but we would expect pathogenic variants in any gene, but obviously in BRCA, to occur much more frequently in cases than in controls. And so what we can do is quantify the relative likelihood of observing each of these sort of factors in BRCA-related versus non-BRCA-related disease. And these will be proxies for the behavior under a pathogenic hypothesis versus a non-pathogenic hypothesis. And so you'll notice that some of these evidence items are the kind of things that you would do if you were assessing a variant classification or interpreting a variant, in a proband. The added power and what makes these so useful as tools, these multi-fact studies, is that they look across huge populations, tens of thousands generally, of individuals, and they can look at multiple occurrences of the same variant. So as we all know, the sort of enemy of variant interpretation for pathogenic variants is that they are so rare, and so the amount of evidence that we can compile for them is limited. But by looking across large populations in a single go, or a single analysis, we can then quantify these factors and these likelihood ratios with much greater certainty and much greater power. So jumping back in time, back in 2007, Doug Easton released this paper while working with Dave Golgar, where they took 70,000 individuals who were tested at, I think it was Myriad Genetics, for BRCA1, BRCA2, for breast or ovarian cancer presentations. And so they had testing of the BRCA1 and BRCA2 genes. They identified 1,400 VUSs among 4,600 probands. And so this gave them a really rich starting point to try and evaluate what the likelihood ratios for each of those factors on the preceding slide were across large numbers of individuals and large numbers of variants. So I'm going to dive into the maths of just one of those factors, and I will say it's not hugely important to understand the maths top to bottom, but this is just a sort of way to illustrate how these kind of models are constructed in practice. And so I'm going to look specifically at co-occurrence. So a lot of the mathematical models that underpin these multi-fact studies have existed I think this co-occurrence one approach was released in 2003 and is still exactly what is used today. So a lot of these are really long-standing mathematical models. And so for co-occurrence, what we're interested in is modeling the expected patterns of co-occurrence of a VUS alongside a deleterious variant if that VUS were to be pathogenic or if it were to be benign. And the mathematical model we use for this is a binomial likelihood model. Like I say, it's not super important to understand the full ins and outs of the equation, but there are three parameters to this model, and it essentially models how likely it is that you have seen the data that you have seen among your surveyed individuals. So the first thing that we have to dictate is the probability of observing a variant in trans with a known pathogenic variant. So we have to estimate that up front, and we'll move on to that on the next slide. And then what we do is for a given variant that we're interested in, we can plug in the total number of times you've seen it and the number of times that it was seen in trans with a known pathogenic variant. And what this equation essentially represents is it's split into two components. The first component is if you have seen the variant in trans with something k times, something pathogenic k times, then this yellow term is the likelihood of seeing it that many times in trans. And then the purple component here is the likelihood of the inverse, so the likelihood that you saw the n minus k non-co-occurrences. So lots of double negatives, but essentially these are the two likelihoods that make up the bulk likelihood, the likelihood of seeing the co-occurrences that you saw and the likelihood of seeing the non-co-occurrences that you saw. So I've just moved the equation up into the top left. And so the way that we construct these two models, one for pathogenicity and one for benignity, is by evaluating or estimating two different values for P. So we need to evaluate how likely it is that we would expect to see two pathogenic variants in trans, and how likely it is that we would see a benign variant in trans with a pathogenic variant. And so that is what differentiates the two models for pathogenicity and benignity, is this p-value. And so what they did was they looked across their cohort, and I'm just looking at BRCA1 here. So the values are different for BRCA2 because the expected rate of co-occurrences are different between BRCA1 and BRCA2. But taking BRCA1 as an example, what they saw looking across their entire cohort, 1 in 10,000 individuals were identified as having two pathogenic BRCA1 mutations in trans, which is sort of unexpected because it's presumed embryonic lethal BRCA1. But that's the value they came up with, which is equivalent to 1 times 10 to the minus 4. So the likelihood of two pathogenic variants being in trans with each other is 1 times 10 to the minus 4, at least in this data set. And conversely, for benignity, it's much more common to see benign variants in trans with pathogenic variants. And actually, if you follow the logic, what you can do is just say that your benign variant is going to be in trans with a pathogenic variant simply at the background rate of pathogenic variants in your data set. So it's not too important to understand, to grasp that now, but that's what the logic is. So they observed that 6.6% of individuals in their cohort had one established pathogenic BRCA1 variant. And so if a benign variant were to be in trans with it, it should be in trans at roughly that proportion. So you construct these two models by simply changing the p-value based on the data that they've collected. And what you can do, again, this maths is a little dense, but you can generate this generalized model for BRCA1 of expected rates of co-occurrence if the variant is pathogenic, which is the top of this fraction, and the expected rate of co-occurrence if the variant is truly benign, which is the bottom. Yeah, so we can do an example variant here. So if you had seen a variant 8 times in total in your data set across 8 different probands, and once it was in trans with a pathogenic variant, you can plug n equals 8 into your equation and k equals 1 into your equation. And what you get is this very low likelihood ratio of 0.0024 towards pathogenicity, which in other words is minus 8 points, which is 8 points towards benignity, essentially. And so this is really strong evidence for benignity, which is intuitive because we've seen the variant in trans with a pathogenic variant. So this is just one example of the kind of maths that's done to generate these likelihood ratios. There are loads and loads of different ways that each of the evidence items are constructed. So you can use logistic regression is very common when you're evaluating the family history of or the relationship between pathogenic variation and family history. And those of you who've used Cool Bayes in your work will have already seen basically in practice the model that's used for co-segregation. So that was all from the first big study, which was Easton et al. In 2007. Since then, there have been occasional releases of updated and expanded data sets. So there are 4 listed here. These are collectively, these 4 plus the Easton paper are the ones that are recommended for use in the CANVIG guidelines. It's important to note that these come around quite rarely because you do have to assemble these huge case series of tens of thousands of probands in order to get interesting levels of signal. And so now I'm just going to speak very briefly about the clinical implementation of these analyses. So this is just a screenshot of the multifactorial data that was collated for a particular BRCA1 variant. And when you are presented with multiple data sources for a given evidence item, I think the instinct is to slightly wince. So you would always wonder, what values should I be using? So for this Parsons data set in the top of the screenshot, you can see that there's both the combined and constituent likelihood ratios. So when we do these multifact studies, you would collate each of your evidence items individually, convert them to log likelihood ratios, and then simply sum them for a given variant. So we need to decide which values to use. Do we use the constituent ones? Do we use the combined one? How do we combine different instances among different epidemiology studies of the variant? So how do we combine the results from Parsons et al. With the results from Lee et al. And under what ACMG code? So this data was not accounted for in any form in the original ACMG version 3 guidance. So we need to know on a technical level what code to apply it under. And so the way that this has been handled by the Enigma BRCA VSIP is to appropriate these to evidence items PP4 and BP5. So historically, these were for phenotypic specificity and alternate molecular basis for disease, respectively. Breast cancer is a very genetically heterogeneous phenotype. So there isn't really a specific genotype-phenotype link to make there. So the 2 evidence items were both utilized by the Enigma VSIP to essentially accommodate this multifactorial genetic epidemiology data. So there's a big long blurb here, but essentially you can incorporate any kind of calibrated evidence type that might include reported family history, breast tumor pathology, case control data. You can also apply it to unpublished data. So if you have any kind of small scale in-house analysis of this variant, if you have multiple pedigrees, you can incorporate it there as well under these 2 criteria. And they do delineate that you assign the weight based on the combined likelihood ratio. So probably seems very intuitive, but on that previous slide where we saw the Parsons data broken down into its constituent parts, you would take the combined likelihood ratio. You can still apply under the Enigma VSIP relevant codes based on the features of your proband. So obviously these studies are based on huge numbers of probands sampled elsewhere. You can still apply relevant codes based on the features of your proband, provided, for example, if you have extra co-segregation data from your families, you would need to ensure that the segregation data from your family exceeds at least 1. So you can't just tack evidence points onto the existing multifactorial evidence unless they are already exceeding supporting. So if you had sort of, 4.5 evidence points coming out of your multifact data, and then you had 0.6 coming out of your in-house pedigrees, you couldn't just add them together to get 5.1. You would have to discard your sub-supporting in-house data. And then I started making this slide expecting it to be a lot more complex. And then I realized actually it's quite simple. So I've got my lab and obviously all the GLHs are members of CANVIG UK. And so CANVIG guidance has become one of the main sources of cancer variant guidance in the UK. I was expecting there to be a lot more differences between how we recommended multifact data to be implemented between the 2, but actually Enigma and CANVIG UK are broadly very aligned. They both say to use PP4 and BP5 for your evidence codes. We both stipulate the likelihood ratio that you use should be the combined one, which seems intuitive. The main area where we do slightly diverge is in how we recommend combining different data sets. So in Enigma, they simply say to sum all of your constituent data sets together and then convert it to an evidence strength. The extra consideration that we make here in the CAMVIG guidance is that you need to ensure that there is independence of samples between the data sets. So if, for example, you had, and we'll see an example on the next slide, but if you had two data sets where the second data set was essentially taking the first one, all the samples from the first one and expanding on it, then those wouldn't be considered independent measures of variant effect because you have substantial overlap between samples in your two studies. If they can be guaranteed to be independent, then you can sum the evidence strengths estimate likelihood ratios as for the Enigma guidance and then convert it to an evidence strength. If you can't guarantee they're independent or they're not independent, then you simply select the data set that's got the greatest number of surveyed families. So there's a lot of notes of caution to be aware of when using multifactorial evidence. The top 2 points here are quite practical and useful. So the top one, if a new study is published or you're going back to an original publication, you have to be really careful on a practical level about how the log likelihood ratios have been calculated. So if they don't give you a raw likelihood ratio, but they give you a log likelihood ratio to start with, these will usually be taken to either base 10 or they will be the natural log. And it's not always clear from the publication which it is. So if you wanted to convert these to the ACMG likelihood ratios, i.e. the exponent points, then you would have to follow this formula that's down here. Sorry, you can't see my mouse, but the equation in the blue box on the right-hand side. You just have to be really careful that you know what log was used to take the log likelihood ratio in your publication. We've had to go back to authors in the past to clarify this. We've had incorrect data. We've made the incorrect assumption and had to clarify it before we could display data. The second point, as we've alluded to, be careful with reuse of data between publications. So there's a landmark publication in 2012 by Lindor and colleagues, but that was an amalgamation of data from previous studies. So trying to, collate existing data into one big data set that obviously does not have independent samples compared to the constituent data set. So you can't use those as independent measures of effect. And then there's some more technical ones, which I won't spend too long on, but there's always issues when you're establishing any kind of model of pathogenicity or benignity. You are making lots of assumptions along the way. So we estimated the predicted rate of co-occurrence of variants in that example that we did before. There will be inaccuracies in that estimate. It will be based on the sample that we took in that data set. It may not apply to other data sets. It's an imperfect estimate with a confidence interval, and none of these multi-fact data sets will apply or display any kind of confidence level around their likelihood ratio estimates. Another big one is ascertainment bias. So particularly an issue for co-segregation. One of the papers, I forget which one, it was the Enigma multi-fact paper, invited clinicians to submit essentially tricky cases, and the likelihood of submitting a tricky case that has convincing segregation data for a particular variant is more likely than submitting a family who have got a less worked up or a poorer sort of level of segregation data from the analysis. So you're more likely to work up families that already have convincing segregation data. And so your estimates of co-segregation contribution to likelihood ratio is likely to be inflated. You've got to ensure that all the likelihoods that you're summing within a multi-fact study are independent. That's not really been an issue with existing publications. I think it's broadly accepted that all the likelihoods you're adding together are indeed independent, but with future publications, it's something to be wary of. And the other big issue is that we assume full penetrance generally when compiling these models. And so your model of pathogenicity is predicated on full penetrance, and so it doesn't really capture incompletely penetrant effects by and large. I will say up front that I have absolutely no connection to ACMG version 4, but just looking ahead to how we might be incorporating this. So like I say, in the original version 3 guidance, this evidence wasn't accommodated, and it was only when the BRCA VSIPs were generating their gene-specific guidance that we incorporated multi-fact data. And so we appropriated PP4 and PP4, BP5. We can imagine a similar picture might be the case with ATMG version 4. So we still have accommodation for phenotype specificity under this lock-phi code. We still have alternate cause of disease under the ClinAlt code. It may be that when gene-specific guidance is released, that we'll similarly see multi-fact data occupying those spaces in the ACMG version 4 guidelines. I have absolutely no inside news on that. This is purely speculative. And obviously, the current version of the guidance is the consensus gene agnostic guidance. And so actually gene-specific guidance that incorporates multi-fact data for BRCA may still be some years away. And so just, I'm slightly running over, apologies. So I'll just very quickly talk about multi-fact data and how we display it on CanVar UK. So this is part shameless plug for the CanVar UK database, which my lab runs and which I maintain. So this is, you may be familiar with it, some of you, but it's a database of all possible single nucleotide variants in 115 different cancer susceptibility genes. So you can search a gene and variant of interest, and we collate lots and lots of different data types, in silico data, for example. We also collate functional data sets, which George alluded to very generously earlier. So on the left-hand side, there's just a snapshot of some of the functional data sets we have. And in the bottom right, you can see all the multi-fact studies that we collate as well. So we have currently 4 of the 5 studies in there. We've had to briefly take one off while we communicate with authors actually about sample independence. That will be back online very shortly, either today or tomorrow. When we display functional data, just as a just for everyone's information, it looks a bit like this. So we display all the functional studies that cover your variant of interest. We also collate some splicing studies too. In terms of multi-fact data, so this the screenshot I put up before is the real exemplar, or the real data for this exemplar variant. So we display all multi-fact data, all multi-fact scores, sorry, and log likelihood ratios that are identified among these 4 and 5 data sets for your variant of interest as well. So I think synthesis and sort of nucleation of all of this data into one place is hopefully an extremely useful resource that saves people going back and forth to different resources. So I think that's it for me. Thank you very much.
Thank you
to take questions.
so
Sorry
much,
for the
Charlie.
slight overrun.
Don't worry. Thank you very much indeed. Thank you very much to both Charlie and to George, because obviously, as you can see, there's a huge amount of work gone into preparing these slides, but complex information to convey as well, particularly for me as a not very good on the old stats. So Charlie, thank you for the statistical analysis as well. I've got a quick slide just to remind you that if you haven't yet enrolled for the online individual assessment for classification of BRCA1, BRCA2 and other HRR genes, so in other words, run 16 of that EQA scheme, then please go to the website to register. The actual assessment will take place across the whole of April, and we will be reporting the results, I think, in sometime in May. And as is per normal, we'll also be having a webinar to go over those results with the cohort of laboratories that took part in the EQA in April. So if you want to know more, please go to the GenQA and EMQN websites. The address is down there, and we will obviously direct you to where you need to go to in terms of registering and participating. Next slide, please.
Simon, can I just jump in there as well? Some news hot off the press as well, just to draw your attention and fits very neatly into George's presentation, is that the GenQA Genie module for somatic variant interpretation was launched just 48 hours ago, and we've had a massive over 100 individuals already complete the module. So I think it's been waited upon for a long time, and everybody's very keen. So it fully supports the SVIG guidelines. So please come to the GenQA website if you need some more information on that, and we'd be great to point you in the direction. There's a free trial on just now, so it won't even cost you anything to try out your SVIG understanding. So hopefully that will boost, we'll see a great increase in the percentages of correct answers, George, after today's webinar as well, hopefully. Lovely. Thank you, Simon.
Thank you, Sandy. Right, okay, so that's the end of today, the webinar, the formal presentations, but really the next section is really over to you. As explained at the start, please do submit your questions to the Q&A function at the bottom of your browser bar. We've got a number of questions already come in so far. So I'm going to kick off with a question I've got a question for George and a question for Charlie, and then I'm sure I'll hand over to Sandy for a few more. So for George, obviously the UK's has a very strong and established network for delivery of genomic services. And in your presentation, you presented the new ACGS guidelines and also their applicability to the SVIG UK framework. What do you see as the biggest challenge in achieving national standardisation with those guidelines and framework, and how would you reconcile the differences in local practice?
Thank you, Simon. So I think when we started the work, the biggest challenge was the lack of a standardized approach, and that's why we're working on establishing these criteria for somatic variant interpretation. So hopefully the first step to help in standardizing the approach across England and hopefully beyond is having very clear and friendly to use guidance. Other than that, I think the training, to help people training in these resources, and that's something we were also aware of at the start of working on SVIG, is to provide training material that people can use. And that's the fabulous work by Chris from the GTAC to create that training material that goes alongside this. Then another fabulous tool is what you guys present with us in GenQA and EMQN, providing these schemes that can help assess people's skills in applying the criteria, and then running webinars and follow-up presentations to help people in understanding what they've done, what could be done better, and so on. So I think these are some of the important ways that we can engage and improve for all of us that pathway of variant interpretation, because also publishing SVIG, we already know of things that we need to improve, change and modify after amazing feedback from multiple labs, not only within the UK, but also excitingly internationally. We had multiple emails asking for certain criteria, questioning various things. So hopefully that engagement, training, testing variants will help us all improve the standards and make sure that we have something that is streamlined, efficient, and correct for the patients.
Fab, thanks, George. I mean, I think we've been obviously running these webinars for a couple of years now. So I think the work we've been doing with you guys and the community in general to try and promote the educational component of variant interpretation and how we take the new developments moving forward has hopefully given you, the audience, a stronger understanding of what we can do and how we can learn together as a group.
And I would say, Simon, sorry, Simon, can I just come in there? I would say also there's definitely hope, because GenQA, we run our first SNVs for germline variant classification scheme back in 2014. So we've now got 12 years experience. And I would say our bubbles on our bubble charts are definitely getting bigger and less variable. However, for germline CNV classification and somatic classification, we're still back at that early stages. But I think George's absolutely right about the education resources, the guidelines. But of course, I think CNVs and somatic variants are much more challenging, because you're not just looking at the pathogenicity of the variant itself. You've got to then layer on the clinical significance on top of that, of their oncogenicity, for example, or your CNVs within the region of the genome. So it is more challenging than just the germline SNVs, I would say, definitely.
No, undoubtedly, I totally agree with that. I mean, we have a question on oncogenicity and actionability. Do you want to drop that one in there for George?
Go on then, and then we'll attack Charlie with his questions. Right, George, so the SFIG framework focuses on oncogenicity classification, and it's used alongside the AMP tiers for clinical actionability. So in practice, how do you communicate a variant's oncogenic classification separately from its actionability? And does having the two parallel systems running risk confusion both in the labs and with the clinicians?
Excellent. Thank you very much, Sandi. So, of course, we want, when we're presenting variants to the clinicians, we want to reduce any confusion to any extent. So just to clarify for those who are less familiar with the somatic language on the code, we have oncogenic classification that sometimes can be followed by actionability classification, which many people will know as the AMP tiering, which was published in by Lee et al. 2017. And so the oncogenicity classification, we're talking about a biological impact. We're answering, is this variant contributing to the tumor development? The actionability question is slightly answering a different question, which is what's the clinical utility of the variant? Can this variant be used to for the oncologist to give the patient a targeted treatment, or they can be used to put the patient in a specific diagnostic pathway? And that can be impacted by other clinical guidance, whether it's international, national, drug licenses, trials availability, what drugs are funded and not funded. So what we do is when we assign oncogenicity, any actionable variant need to be oncogenic before it can be actionable, while not all oncogenic variants are actionable. And the common example of that is P53, that you can identify a oncogenic variant that doesn't have any specific treatment to be done. When we report the variants, we don't add, at least that's a practice in the UK, we don't add another layer of actionability as in a tiering. In most instances, we report back with a statement on this may impact specific line of treatment. This may make the patient or this tumor sample more sensitive or increase the likelihood of response to a certain treatment and so on. So we do relate where clear clinical framework is available. We do relate to the possible impact on patient treatment. And when the variant is oncogenic with no clear implication, we just put a generic statement that this could be used in clinical management or clinical trial selection if available. I just want to note that we are working on another initiative, hopefully nationally, to try and create again a more up-to-date standard that can be help in reporting actionability alongside oncogenicity.
Great, thank you very much. It is complex, isn't it? But the mist is clearing now, I will say.
And we've got one more question on for George, I think, before we can move on to Charlie.
Okay.
So George, I've got a question about the B2 standalone VUS criteria, and whether it's possible to come across a situation where the variant didn't fit the model of action for the gene, but might be enriched within the tumor data under 04, for example. Would you continue to interpret and see whether other elements might be available, or would you stop because B2 is a standalone code?
Thank you very much. I saw the question, Mary. Excellent question indeed. So it's a question that we thought about a lot when we were doing the B2 criteria. And as I said in the talk, the most common example of these is seeing a truncating variant in a gain of function. So you see a nonsense in the middle of EGFR or KRAS. That still would go for us as a VUS, even if the variant was present in the enriched in certain tumor databases. That will still be a high level of conflicting pieces of evidence, which will still come under many criteria as a variant of uncertain significance. I've not come across many like this, but the only example that I could think of is sometimes common or variants in P53 that you see few times in tumor databases, but you have strong pieces of functional evidence pointing towards no impact on the gene, and you end up again in a VUS. So you have to be careful, because of course there can be canonical splice variants that results in exon skipping, like with MET exon 14 skipping. So people have to really understand the mode of action and the impact of the variant before they go towards using B2. But I don't think necessarily being in variant in tumor databases would steer me away if biologically the variant does not make sense. We know that tumors are messy, and you can see a lot of somatic variants all around the tumor. So you could still see these variants even if we don't think the mode of action fits. I hope that answers the question.
Thank you, George, very much indeed. Charlie, we're going to come over to you, I think, next. You haven't got away with it. Don't worry. I'm going to kick off with a question on sort of clinical communication and with relating to the more complex multifactorial models. I mean, clearly there is going to be a layer of complexity coming into the interpretation realm when the ACMG version 4 guidance come on board. Do you have any suggestions for how to explain the classification based on statistical evidence? So the data you, for example, presented on likelihood ratios in a clear, clinically and meaningful way for the end user?
Yeah, I think any time that statistics meets clinical use, there's always a horrible car crash interface. I think the way that the terminology that we've used, like I said, there were about 3 or 4 different phrases that we use for odds path, likelihood ratio, Bayes factor, which that kind of lack of standardization doesn't help. I know there is a push to actually move away from the phrase odds path, which is probably the most dominant phrase, to something a bit more meaningful. I actually think that the earlier publications, so the way that it was described in publications like Easton et al. Back in 2007, just a phrase like odds in favor of neutrality or odds in favor of pathogenicity are probably as clear as you can get in terms of communicating to clinicians. Any kind of phrase, because those are presented as sort of ratios to something to 1. So the odds in favor of pathogenicity are 350 to 1 based on, and then lists the constituent evidence in brackets. I think that's probably as clear as I can think to make it. But yes, I think we've strayed gradually further and further into very jargon heavy territory, and I think returning to something that's a little bit more familiar in terms of just
it's going
odds.
to be interesting for us as the quality assurance providers, because we obviously see the end reports from all the laboratories, and as we start to see the implementation of these types of models in the variant interpretation, we'll start to see the impact on the way laboratories convey that information to the end user. So I think it's going to be challenging for us as well as how we assess it, but also challenging, educational with respect to how laboratories address that issue and the complexity of it. I mean, obviously the end result is you want to have a clear message to convey to the end user, and maybe some of this complex stuff will get buried a bit more further into the fine print of the report. But I think it is going to be challenging to see how it comes out in terms of how to take that complex data and convey it in a meaningful way to the end user. I guess it's a watch this space type scenario, isn't it?
Yeah. I mean, a lot of these studies have been sort of implemented for quite a long time. My hope would be that under version 4, I don't think we will see too much change in the way that these are actually implemented themselves. I think if we were to want to standardize it and come up with a clearer, more systematic way to describe it, I think now is as good a time as after version 4 comes out, because I don't think there should be vast changes, again, with absolutely no inside baseball knowledge, that these are going to be, I don't think these are going to be implemented too differently after version 4.
Great. Thank you very much indeed. Sandi, over to you.
Yeah, thanks, Simon. So, Charlie, another question, and this actually resonated with me particularly on your slide where you had all the sort of the things that you need to have to think about at the back of your head, and that's about the problem of people using the same information twice. I mean, we see this with right across the board, and it's really difficult to know whether studies are unique patients, not just the data sets, but whether those patients are being analysed in by multiple sources. What do we do about that?
It's a great question. I think that sometimes we have the benefit of geography. So a lot of these studies have come from very disparate parts of the world, and the recruitment
Mm,
was through completely different agencies. So there's a French study that comes from the French health system. The Enigma study came from,
sure.
I've drawn a blank, I can't remember where the Enigma study came from. But the American studies come from commercial testing laboratories. Um, what we have found is that actually contacting the authors of the paper is very useful, and that they're typically very responsive because they're interested in ensuring that these data sets remain in clinical practice. So
Sure.
I think I mentioned in the CAMVIG guidance we have established that four of the data sets are almost almost certain to be completely independent of one another by contacting the authors and confirming that participants were recruited from completely different probands were recruited from completely
Mm.
different systems, be it different healthcare systems or different countries, for example.
Quite time consuming and capacity heavy, isn't it, to be able to do that?
That is true.
Mm,
I mean, from our end, it entails an email. I think from their end, it's obviously the burden of the work is on them to just
sure.
cross-check with each other that nothing has overlapped. The yeah, the study that I mentioned that is CAMVIG recommended but not currently on the Canva platform was exactly that problem because that was
Right.
a French paper that was led by a key author on an earlier GenEpi paper. So we knew there was overlap in the research team that were conducting the studies, but she was able to confirm that recruitment was entirely separate. I
Okay.
guess you can't 100% guarantee that
Mm,
related probands, albeit probably distantly related probands, could have been recruited in different settings. Um, there is unfortunately always a slight level of uncertainty. Um, yeah.
yeah. So the key is contact the authors, because if you can get evidence from them, then it makes it you're more confident, I guess, that it's standalone data.
Yeah. And I mean,
Great.
so my colleague who mans the CamVig inbox will be angry at me for saying this, but you can always email CamVig and just double check because we do sort of or they do sort of steer on this kind of thing and ensure that
Sure.
data set independence is as solid as possible, or as robust as possible. So if you
Great.
have specific queries about pairs of data sets or you identify a new data set and are curious, then CamVig can also potentially, the CamVig team can look into um, contacting the authors themselves.
Right. So great advice, but we won't disclose that you said that
Yes, yeah.
to
It
them,
was
to the
I,
team.
I will
someone.
tell Sophie.
Brilliant, thank you.
Thank you.
Simon, I think that's all our questions answered, which is fantastic.
Yeah, it is indeed, Sandi. Absolutely. Over to you then.
Well, thank you very much, everyone, for participating today. We hope that you found it useful. I've learned a huge amount, I must admit, although I wouldn't say I'm a bioinformatician in any shape or form. But thank you to our speakers. Do we have a next slide for our yeah, no, I think we'd like to say thank you. Also, a quick reminder to do complete the survey, because as this webinar really was a product of feedback from participants from previous webinars. So please do let us know what you would like us to focus on going forward. Huge thank you to Charlie and George for a really, really educational and great resource, and for taking the time to be with us today. And a big thank you, as always, to Simon, my co-chair, to Angie and the GenQA EMQN team that obviously run the Genie variant modules, and also the team behind everybody, and AZ and MSD for their funding. So a huge thank you, and take care. Thank you. Bye-bye.
Good afternoon.